Hi, everyone. Thank you for this opportunity today. My name is Miwa, as she explained. I work at CSIRO in Australia. CSIRO is the Australian Government Scientific Agency. So today, in one of the two talks I'm giving today, this one I would like to introduce about IMOS eDNA sub-facility. IMOS is an integrated marine observing system. It's a long-term monitoring program in Australia in the marine field. And this IMOS has been operating for almost 20 years, but this eDNA sub-facility was recently launched only 6 months ago. So we're going to add a whole new layer of Australia's marine biodiversity monitoring, which I'm really excited about. So I'm going to tell you a bit about IMOS now. IMOS is funded by Australian government, enabled by the NCRIS. It's operated by consortium institutions that you can see there, and the lead agent is the University of Tasmania. So why was IMOS established? It's because oceans matter to Australia and, of course, the global communities as a blue economy. And in order to sustain this valuable resource that ocean provides to us, we need high-quality, sustained marine and climate data. And in order to provide those data and information, IMOS was established in 2006. And since then, it's been delivering high-quality marine data. IMOS data contains a range of physical and biological data, and all the data are open, usable, reusable for anyone on the planet. So it's freely available to scientists, students, industries, and any other stakeholders for any use. So it can be for research or educational purpose or operational activities, and then anything that will support assessment of ocean health at the global and then local scale. So what kind of samples and data are collected within IMOS, and where are they collected from? This map on the left, yeah. So these are the national reference stations. There are 8 stations in operation now. These are not only the sites that IMOS operates, but today I wanted to highlight these 8 sites because these are the places where eDNA samples are collected from. And also this longevity, long-term history of these sites are quite impressive. So this, when I say long-term history, it goes more than beyond the IMOS operation, which is 20 years. So some of the sites down south, Port Hacking, Maria Island, and Rottnest Island, they were established in 1940s, so for about 80 years. And at the same time, when those NRS were established, they started all sorts of data collections and sample collections. So what types of data? These one on the right-hand side, so they shows some of the key data streams at NRS, not all of them. So you can see some physical and biochemical data schemas. So those are temperature, salinity, oxygen, carbon, nitrogen, current, or you can think about. And then there are also biological and ecological components. Those include phytoplankton, zooplankton biomass, microbiome community data. So you can see this very comprehensive and full color pictures of ocean pattern here, which is amazing, but there's something missing, and it could be even more amazing if we could fill that gap. And that gap is that it doesn't measure larger organisms that most people think about when we think about marine biodiversity. And this is where we are going to change with this new eDNA sub-facility. So this eDNA sub-facility was launched in July last year. And so it's sitting under biomolecular observing facility. There are a number of facilities under IMOS. It's a really big program. There are, I think, 13 facilities operating now. And eDNA sits under this biomolecular one. There's also microbiome ones. They have been operating for a lot longer, and I'm going to explain more about this now. But this is just how the facility and the sub-facility structures are like. And our aim is to leverage these existing samples from marine microbiome programs and then expand this long-term monitoring to macro, larger organisms. So this marine microbiome initiative I just mentioned, this one is led by the Australian Microbiome Initiative, and they collect all sorts of samples, not only from marine, but also from freshwater, terrestrial environment. And so on the left-hand side, you can see the map again of the NRS, National Reference Stations, with the years. So that's when these marine microbiome samples started the collections. So 2012 on the East Coast, and then 3 years later, they expanded over on Australia. So we have these samples from 2012 at monthly basis, from surface to the bottom at multiple depths, which is really amazing to have that access to it. They collect 2-liter water samples at most of the sites monthly basis, but 2 sites in Darwin and Kangaroo Island, they collect every 3 months. And then they apply 3 assays, 16S bacteria and then archaea, and then 18S eukaryotes at this moment to understand some microbiome communities. And all the standard procedures, the protocols and data are fully available, free, open access. So I want to mention about this open access bits about this project. And so what's amazing about this initiative, this marine microbiome initiative, is not only about this longevity and the consistency of the operation, but also how they are really committed to make everything transparent. And so their data and protocols are accessible through this Bioplatforms Australia. So this web interface, you can get the data depending on the taxa of your interest or regions or assays, the amplicons, and all sort of things. And then, but their data doesn't stop here, the data stream. So they're also making the data available and publishing the data to these international databases. So raw data are available from NCBI, and then OTU table, taxa information are available in GBIF. And if it's a marine data, they also go into OBIS. So they are really conscious about, this international data architecture, and then they want to make sure their data doesn't stop just within the Australia or IMOS program, but also they want to make sure that their data are interoperable and reusable across the international eDNA communities. This one, I'm just going to go very quickly. This is just some of the examples how the data are used. And one of the examples in the top left is the Australia State of Environment. It's a government report. They, in the marine sections, heaps of IMOS data have been used. And there are a number of publications. People are using this IMOS data to really understand the ecosystem within Australia, but there are a couple of examples on the bottom. That's a global biodiversity paper, and then IMOS also contributed in that. This is another type of samples collected at NRS, and this is not within this biomolecular section, but I would like to highlight this because we are also going to use this data in the metabarcoding. So in each sampling time on all these national reference stations, so mostly at monthly basis, they drop 3 nets. 2 of them are used for morphological identifications of plankton, and then all this abundance and biomass are recorded. One of them are kept for bulk metabarcoding analysis. And some of the samples have been already analyzed, and then they were analyzed by PhD student, Tina Berry, and she published these couple of papers, and she really revealed really cool patterns like seasonal variations and some abnormal seasonal patterns associated with the marine heat waves, and also identified some invasive species, endangered species. So this kind of example really shows that the power of profounding the real ecological patterns in the IMOS data. And these are the type of data that we, as an eDNA sub-facility, are aiming to generate at the larger scale. So I spent a fair bit of time explaining what has already happened, and that's because we are going to build upon those existing foundation of IMOS, and that's why I wanted to spend a fair bit of time on that before I get onto this main topic. So this eDNA sub-facility, we're the team of 4, but of course, these are the main team, but then it's a big collaboration with a number of people involved. So the aim is to build upon those existing IMOS samples, as well as the future ones that are continuously coming in, to expand the long-term monitoring to macroorganisms. And there are a number of opportunities we can think about. So we're going to have a huge data set across the space and time. And then all of those data will come with very, very rich metadata. So IMOS collects all sorts of data, as I mentioned. So all of those are going to be accompanied to this eDNA data, as well as the community data as well, so such as planktons and microbiome stuff. And with all this data combined, there is an opportunity to run really cool modeling studies or distribution to understand distribution patterns. We can maybe use that for like a biosecurity monitoring as well. And for this plankton biomass data I mentioned, I think there's a huge opportunity to really tackle down the quantification part of eDNA, because this is one of the limitations that eDNA has. And but this data is going to come with this actual biomass and abundance data that was morphologically identified. And then another opportunity I want to highlight here is we're going to have a quite good species identification accuracy thanks to the NBDL. That's a National Biodiversity DNA Library project, and they are basically trying to sequence every single organism in Australia. And they recently released a fish database. That was the first one they launched, and they sequenced about 2,500 species, fish species, and a whole genome, and they are available now. So we can use the database and then have a quite good species identification accuracy. And their database, the data, the reference library is all available for anyone to use. And then, as you might know, I'm very passionate about fair data principles, and we have also an opportunity, of course, to make our data available. Well, not really opportunity, but we are going to do that. So there, as we learned in the last few days, and there are quite few like amazing eDNA long-term monitorings around the world, and ANEMONE is one of the great examples. And I think it will be amazing if we can really join our forces together across those multiple projects to make our data fair, and then we can make a big global database together. So when is this data going to come alive? You might be questioning. So this is our milestones that we've been working towards, and as you may know, everything takes longer than we expected, and that's been happening already for us, but this is something that we're really aiming. So up until now, we've been engaging with end users and eDNA practitioners to understand what kind of use case we can have in the future once we have the data available. How would people want to use it? So we want to understand, for example, like interest of taxa and what kind of assays people have been using, what's working, what's not working for them. And then based on those engagement outcome, we are establishing protocols now. So for this protocol establishment, we're not looking into, we're just looking at the post-PCR part, because samples already collected and then extracted. So for PCR step, one of the important things is that what assays we're going to use. And we have a resource to use about 3 assays at this moment. Hopefully we have more, we can apply more. But there are so many options. There are so many different assays. So we have these 5 in our mind now. We're running a pilot study to see what could work better for our case. And then we're hoping to select 3 of them, of those 5. So if you have any experience using those assays, and if you have some insights, advice to us, please let us know. I would like to hear. This is the land of my fish, and I'm sure some of you have tried multiplexing E and U, and this is new to us, so I would love to hear any troubleshooting, trial and error that you might have done already, so we can skip that step. Then once we establish the protocols, then we want to process the samples, the actual IMOS samples. For this pilot study, we collected extra samples just for that, but we're planning to process this existing amazing samples once we have the protocol set. Probably not the whole 15 years of samples in one go. Maybe we're going to do the first couple of years, the most recent ones first, and then analyze the data and see how it goes. But this is our plan. So I put please use our data here because I would really like to highlight that it's important to understand that this IMOS is a data initiative, and within this project, unfortunately, we don't get to actually analyze the samples and then turn them into real science. So we're funded to produce the data for other people to use, and then for them to use and then turn them into fun science stuff. So data use cases are really important for us to demonstrate the impact of our project, and so that we can continue getting our fundings, not only to sustain my career, but also, more importantly, to continue producing this valuable data set. So once data is available, please think about the way we can use, maybe PhD students or someone can use it, or even government reports can use that. So yeah, I would love to hear what you think, if there's any potentials that you can think about for this data use cases. And then my job is really to make sure that data is going to be as useful as possible for broad audience. So I would love to hear your comments on that. All right, so this is the last slide. So take-home message, we are leveraging existing IMOS frameworks and samples, and currently we are establishing PCR onwards protocols, and there are lots of exciting opportunities coming up, and we want the data to be used, so please reach out anytime. Here's my email address at the bottom, and I would like to thank all the collaborators to get to this point. Thank you very much.
Oh, thank you so much, Miwa-san. So we have some time to take a couple of questions and comments. Anything from floors? Right.
Hi, thank you. Amazing initiative. I would love to have something like that for Malaysia. I was wondering about data access. If it's all open to everybody, is there any consideration about rare species or threatened species or endangered species? For Borneo, that's always a big topic about making data available to everyone about where you find the rare orangutan, the rare hornbill, the rare fish species. So is there any consideration that you might want to hide some of the data so people can't go and poach them? Or?
Oh, hiding. I thought you wanted to highlight those findings, the other way around. Yes, that's a great point. At this stage, I wasn't thinking, so thank you for raising it. And so I was thinking, making everything available. And of course, through other projects that I'm doing about making data available, I am aware about these concerns, and then people can definitely not openly share everything. So any endangered species that are in risk of poaching, we could do that actually, and then we should probably make a list of those species, and then not just within Australia, but if it's in concerns for other countries, like adjacent countries, then yeah, that would be really important to have that kind of list and then what other people are concerned about, so we can manage it properly. Yeah, thank you.
Okay, any other questions or comments, whatever? Okay, Defin-san, please.
I think slightly related. I wonder about how would you define fair? Because it's like, of course, the right of the firstborn. You have your first data, and of course, you want to have a published and high-impact journal and stuff like that. So it seems like you're giving away the data, and then so what is the catch? That's something that I would like to know. So if it's and also another thing is, of course, this is Australia, and of course, being from the other part is from Indonesia, and of course we also have this data that we do not want to open. So, and also you mentioned about all the samples being collected in Australia. I believe probably some samples also collected from Indonesia. Would you have this political problems in the sense that when you try to use the data, to publish the data, when you said doing the real science behind that. Because I think many of the members in Japan's side are actually evolutionary biologists, taxonomists, and molecular evolutionists and stuff like that. So rather than doing politics, we're more concerned about first authored paper and last authored paper. Thank you.
Okay. Yeah, really great point. And so, as I mentioned, this IMOS role is really to provide the data for anyone to use. So we have no concern about if we, share the data and we haven't really made any publication out of it, what do we get out of it? That's not our concern. So we will make it everything openly available. But I do understand that, some people like academia, taxonomists, and then, some people in the different countries, they have different concerns and they don't want to share the data. That in that case, that's also respected. That's how it is. And then I think it's okay. There would, more communications and discussions are needed, I think, in this space to really make sure that we're on the same page. But if that's your concern, I'm not sure if I understood your question and I'm answering properly. But
You're answering properly.
yeah.
Very well.
Okay. Thank you. Yeah. So I think that's how I see from the IMOS view, and then also another project that I'm doing, the FAIR eDNA view.
About standardizing protocols. So I think everybody has their own different protocols. So mine is better, mine is better, mine is better. What is happening, I think, in the field. So if you have any insight on how to basically push this one protocol is the best one or something like that.
Protocols as is like data publishing protocols or before that?
Data collecting protocol.
Collecting samples and then processing. So maybe Christian can talk more about it. He's really expert in the standardization. But in my view, I think
because we believe anemone is the best.
Yeah, so there are quite few regional best practice protocols, like Japan has one and Australia, they developed one as well. So I think using those and then cherry picking the best parts that could be applicable for your case, that could be the best way to proceed. But I do also understand that we cannot just have one best protocol that should apply to everyone in any regions, any environment. I don't think it's not possible. So some flexibility is required when we talk about standardization.
Thank you.
Thank you very much.
Okay, thank you so much, Miwa-san.
Thank you.
Yeah, thank you.